From here, I'm going to link up to first order of such questions so that we are done with this investigation questions, then we can carry on other things. Because clearly you're going to have investigation questions. I was saying, when they ask you for your dependent or independent variable, first you look for the aim of the investigation. And in the aim, you are going to find your dependent and independent variable. So in this case here, muscle protein synthesis is dependent on the amount of protein taken in daily. The dependent variable is muscle protein synthesis, and independent is amount of protein taken in. We get it from the aim. I'm going to give you another question paper and you'll tell me where are the variables. Give two planning steps that the investigators would need to follow. What are the planning steps here? Hmm? Okay. Generally, planning steps, first thing you would say they will have to get permission from the individual. If you are dealing with humans in your investigation, always getting permission from participants or doctors would be, it's a medical kind of investigation, would be one of the planning steps. If you are dealing with large animals, starting with things like, let's say, if you are dealing with animals starting even from rats upwards, you have to get permissions from departments that are involved with animals that say that you should not treat animals like you're using them to just have fun in by killing them. You have to get permission from such bodies. But if you are dealing with things like bacteria, you don't need permission. If your investigation is about bacteria, planning steps, you don't mention permission. Now, you will also have to determine you have to decide on what method you're going to use to run your investigation or to collect your data or to collect your sample. Method of that you will use for the investigation or method to collect data, decide on method also to decide on the type of apparatus, the type of equipment you are going to use. You are also going to decide on the duration of the investigation. You are also going to decide on the place of the investigation, the time when the investigation is going to take place. Those are the planning steps that you need. Now, next, give two reasons why this investigation can be considered reliable. You check again. Two reasons why it can be considered reliable. I gave you one already. And then somewhere I pointed out another one and I said we'll discuss it later. So, two reasons why we can call it reliable. Okay, lots of time. The first reason is because they used 100 participants. Look here, I told you, when I say 100, it tells me that this is a large sample size. Large sample size means reliability is high. It is reliable because they use a large sample size of 100 participants. The second reason is because they compared their results with two other investigations that were done, and the results were similar. It's stated here. These results were similar to two other investigations carried out. There are two ways of increasing reliability. I told you, one is to increase sample size. Two is to repeat the investigation. If these people have done their own investigation and then they compare to two other investigations that were done and had similar results, it's as though the investigation has been done 3 times already. So as far as repeating the investigation is concerned, it has been done about 3 times already. That makes it reliable. Calculate the percentage increase in MPS from 20 grams to 40 grams. Percentage increase from 20. So we are looking at from 20 to 40. The values that we are looking for at 20, MPS is 11. At 40, MPS is 12. Percentage increase, therefore, would be 12, that is the highest value, minus 11, that is the lowest value, divided by 11. Why? Because 11 is where we are coming from, and they ask us to calculate percentage. We are going to multiply by 100 over 1. If you do that, you should find the answer. So, in a nutshell, when you are doing such calculations, the top, the numerator here, you just do your subtraction, highest value minus lowest value. Our problem here is usually the denominator, where we don't know whether to put 12 there or what to put the total or whatever. I just summarize by saying 11 is where we come from. You divide by where you're coming from, and you multiply by where you're going to. Your question asks you to calculate percentage. So percentage should be the final expression of your answer. The final expression of your answer is where you are going to. That's why I'm multiplying with 100. And I'm dividing with where I'm coming from. So you can do the math. You have a calculator, you can find out that. Using the data provided suggests why the scientist's conclusion stated that 20 grams of protein was the optimal amount of protein needed daily for increase in muscle protein synthesis. So they're saying 20 grams is the optimum. Okay. Optimum, optimum doesn't mean maximum. The maximum here is 40, but the optimum is 30, 20, sorry. So that's saying why is 20 the best amount of protein to be used if I want to achieve greater results of muscle protein increase or synthesis? 20 is the best amount, the best quantity of protein to use if I want to get the best result. Because when I look here, for example, I gave this person 0. From 0, it was 5. Then after giving 5 grams, the value increased only up to 6.8. That's a difference of 1.8. After giving 10 grams, the value increased only up to 8. That's about 1.2 increase. But after giving 20, it increased by 3 units, up to 3. 3 is much bigger than what I've ever achieved here. Now, I felt that, okay, the higher the amount of protein, the better the result. What did I do? I gave up to 40. But when I gave for 40, from 20 here, it only increased by 1. So the highest increase was when I gave 20, it was 3 units. Therefore, 20 is my best quantity if I want to get the best result. 40 is just a wastage because it's not giving me much. It's giving me just 1 extra unit. So it's a waste to give 40. That's what optimum is about. Okay, quickly I'll share another one here. Let's say here an investigation was conducted to determine the effect of zinc supplement on the levels of testosterone in the blood of weightlifting males. Then we have the procedure here. 60 healthy weightlifting males. Again, the moment I see 60, it's a large size. It tells me that this is reliable. That's reliability. Large sample size. Of the same age tells me this is validity. Were asked to participate in the investigation. They were given the same diet again tells me this is validity for 6 weeks. 6 weeks, all of them experienced it for 6 weeks. This is again validity. They were given same diet for the same amount of time. Their testosterone level in blood were measured every day for the 6 week period. They were given a zinc supplement with their diet every day for 6 weeks. The testosterone level in blood were measured every day, again every day. The average levels of testosterone in blood were calculated before and after the zinc supplement. Average free testosterone per group before and after the investigation is given below. Again, this is reliability. When you do different investigations and you consider the average, means that you are making your experiment reliable. So, identify in this investigation the dependent and independent variables. Please, can someone at least impress me? Which one is the dependent variable? Which one is the independent? Hello?
Blood of weightlifting male.
Say again.
Blood of weightlifting male.
The what?
The blood of weightlifting male.
Okay, Mr. Massey, almost there. Anyone else? We're just missing something there. What is the aim of this investigation? I told you, when you are asked for the dependent and independent variable, the first thing you do is you look for the aim. What is the aim of this investigation?
To determine the effect of zinc supplementation on the level of testosterone
Thank
in
you
the
very much,
blood
sir.
of
Good. That is our aim there. To determine the effect of zinc supplements on the levels of testosterone in the blood of weightlifting males. On the level. So our dependent variable is level of testosterone. Our independent variable is zinc supplement, amount of zinc supplement. That is our independent variable because the level of testosterone depends on how much zinc supplement the person is taking. Are we clear? Hello? Are we clear?
Yes, sir.
Okay.
Yes.
Describe the control that was done for this investigation. What was used as a control?
measuring the testosterone level
Say again.
measuring the testosterone level in blood.
Measuring the testosterone, whatever. Let me see here. Mm, I didn't send that. Okay. The control that was done here was to calculate the average level of testosterone before, look here, average level of testosterone in blood was calculated before and after the zinc supplement. So the control that was done here was to find out how much testosterone they calculate before they even went on zinc supplement, so that from that control, they can say, well, after they calculate the amount of testosterone produced after the supplement, they can use it now to compare the before and the after. Controls are used to compare the results that we obtain after an investigation, or they are used to validate an experiment. So when you calculate values before the supplementation, because you are testing the effect of that supplement. So when you get the values before the supplement, then at least you can use it to compare if the supplement is working or not. So that is your control by measuring the level of testosterone before the supplement. That's your control. Set two ways. So set two variables that should have been considered about the zinc supplement to ensure the validity of the investigation. When we talk about validity, we want everything to be the same, except for the factor that is under investigation. So what can we do? They're asking you here, what can we do about the zinc supplements to ensure that the investigation is valid? So you go back to your text here, the procedure. They were given the same diet. 6 weeks, their testosterone levels were measured. Da, da, da. They were given a zinc supplement with their diet for 6 weeks. The testosterone level were measured, the average level of testosterone. So about the zinc supplement, what can we do to make sure that the investigation is valid? For validity, if you don't use the word same, just consider it wrong. When you talk about validity, we want to see you use the word same. If you don't use the word same, consider your answer wrong. So in this case here, I will talk about probably the same brand of zinc supplement.
Same female.
Say again?
Same same female weight and age.
No, they are talking now about just the zinc. They said the restricted use. With respect to the zinc supplement, what can we do to make it valid? So your answer is only about the zinc supplement. I'm glad you are saying what you are saying though, because that could be another aspect. I will just reach and then discuss that one later. But based on the zinc supplement, it could be the same brand because you could have different brands. Aspen, Claymore, any other different kind of brands that you can have. They might not have the same concentration of zinc supplements in what they are selling. So same brand of zinc supplements. Next, same amount of zinc supplement. Remember, realize that here, I think it was done on purpose so that they can ask this question. They said they were given a zinc supplement. It could be any. So to make sure that it is not just any for all of them, we have to keep it to be the same brand. Secondly, they said they were given a zinc supplement with their diet for 6 weeks. They do not even talk about the amount of zinc supplement. It means that they can give for one person 1 milligram, another person they'll give it 20 milligrams. You will see that the investigation is no longer fair. Not fair is not valid. because probably the person who gets 20 milligrams will produce more testosterone than the person who is getting 1 milligram. So if I need to consider the effect, then I have to give them all the same type of or the same amount of zinc supplement. Now, if they had just said in general, explain how this experiment could be made valid, then yes, you can say the males must be, the participants must be of the same age. Remember, I told you for validity, you must always use the word same. Same age, same gender, but we won't talk about gender because I think they already emphasized that they were males here. They didn't talk about the gender, you will say same gender, same weight, must be of the same weight, same race. I'm not going to use blacks versus Indians versus whites. We want to keep them all at the same rate so that you won't say that based on race, there are other factors that cause some to produce more testosterone than others because of their different race. So they must all be of the same race, same age, same weight, and the supplements must be the same, other things be the same. Write a general conclusion that can be drawn from the results obtained. So these are the results here. Before administering the zinc supplement, they were only producing about very few amounts of testosterone per meal. And then after the supplement, the amount of testosterone increased. So you go back first to your aim. Investigation was conducted to determine the effect of zinc supplementation on the levels of testosterone in the blood of weightlifting males. So your conclusion will be zinc supplementation increases the level of blood testosterone. That is your conclusion. Sexual functions of testosterone in male reproduction. That is your paper one. Okay. Otherwise, it causes the development of male secondary sexual characteristics and also encourages sperm production and development, sperm maturation as well. So the last one here on investigation. An investigation was done. Compare the reflex actions. The reflex actions in newborn babies with premature babies. The ones who premature, early born, and not carried for the full term. A newborn baby's chances of survival is evaluated by a doctor and scored on an Apgar scale. The heart rate, breathing rate, muscle tone, color, and reflexes are used as criteria for this test. So, just from there, please identify the independent variable and the dependent one. Independent and dependent variable. Sorry. Independent and dependent variable. Our aim is here, guys. Investigation was done to compare. That is our aim. From here, you look for your variable. I won't go through the whole question again. I'll ask a last one again for this variable until I'm sure that we are fine with it. Our aim is here. Investigation was done to compare the reflex actions in newborn babies with premature babies. Now, sometimes your independent and dependent variables cannot be so straightforward. You have to make it up somehow. So, my variables are reflex action, and babies, premature, and newborn. Whether it's newborn, whether it's premature, all what I see that joins them together is age of baby. Okay, age of baby. So this part that says newborn babies with premature babies, I summarize it as age of baby. The dependent variable here, therefore, will be reflex action. Reflex action or amount of reflex action or time of reaction, because the reflex action, I can also talk about it in terms of reaction time, that is time of reaction time, and age of baby. So reaction time here depends on age of the baby. That's what I'm saying. Sometimes you need to twist your variables a little bit to understand the context in which these variables are stated out so that you make them sound much better. So my dependent variable is reaction time. My independent variable is age of the baby. Then the last one on this side, this one here. Please, I need people to at least tell me now for this. The male fiddler crab has one normal size claw and one giant size claw. Photograph B. Female fiddler crabs have two small claws that are the same size. There is a large size, there is a range of size of giant claws in the male fiddler crab population. The male waves the giant claws to attract females during mating season. A researcher investigated the importance of claw size for males for successful mating during breed trials. What are the variables here? Which one is the dependent and which one is the independent? I've given you your hint here. This is your aim already. This is your aim. So tell me which one is dependent, which one is independent. Even if you can't understand it now, please, one thing that I need you to remember is that for your dependent and independent variables, you look for them in the aim. Look for your aim first, then you can get your variable. So which one is the dependent here?
Size of male.
Say again.
I have learned.
I'm waiting.
Size of male.
What size of male? You're missing something there. Just before size, what do you see?
Yeah, my say dependent or independent.
I want the, okay, let me start with this. Give me the independent, please. Give me the independent.
No size of male.
Thank you. Just the claw size.
Yes,
Okay, claw size. Claw size, that's your independent, correct? And which one is the dependent? Which one is the dependent?
Mm.
the increase of
Thank you, Mr.
your
Martin. Success of?
male sex.
Thank you. Independent is mating success. These guys need to weigh their claws. If the claws is big enough, it will be very visible by the females, then they will be attracted. So the larger the claw size, the more successful they will be in attracting the females. The larger the clause size, the more mating success they will have. That's what it means. So the mating success depends on the clause size. Earlier, I asked you about the independent variable. I was trying to make it a bit difficult. Guys, to find the independent variable is a bit tricky. So when I'm asked the independent variable, the first thing I do is to rather look for the dependent variable. That one comes easily. The moment I get the dependent variable, it can easily point to me which one is there for the independent one. That way, I get my independent variable more peacefully. But it's not always easy to figure out which one is independent. That's just a trick you can use. To ask you for independent, you look first for dependent. And when you get it, you will now be able to see clearly the independent one. Then you take it as an answer, you write it down. Okay, I'll stop here for the investigation. We left with about 5 minutes. So let's look at this question here quickly. These three sources of variation in a species. Three sources of variation in a species. They could say in a species, or they could say in a population. The same thing. The same thing. So your sources of variation are crossing over,
Crossing.
crossing over during prophase 1, 2, random arrangement of chromosomes at the equator during metaphase 1 and 2. All of these two, you can say meiosis. It will still give you that mark, meiosis. Then the next one is mutation. Sorry that I'm taking the show because of time. Mutation. Another one is random fertilization. Then one last one is random mating, because people or organisms interbreed at random with different partners, producing offspring that are from different couples. Explain why the above. So we have diagram here. We see first there is a population. The population produces more individuals than can survive naturally. The individuals of the population have many different characteristics. It's already Darwinism that I'm seeing here. Some individuals survive longer and produce more than others. The traits of those individuals that survive and reproduce in the population will become more common. Explain why the above example represents spontaneous variation. The reason here is because there are just two phenotypes, two distinct phenotypes. The color of these animals, these are deer. The color of these deers is only either black or white. Two distinct phenotypes. Next, also, there is no intermediate phenotype. For example, if there was an intermediate one, it should have been gray. There is no gray. Gray would be halfway between black and white. If you mix black paint and white paint to give you something kind of grayish. So because of this, this is not continuous variation. Continuous variation has intermediate phenotypes and has a large range of phenotypes. Has a range of phenotypes. Okay. Okay. I'm gonna jump a bit. I just want to go and look at this other question. What's happening here? Cloning. Name the process shown in the diagram above. This is cloning. Give two possible benefits of this process. One is to increase food security. Two is to prevent extinction. Three is to produce organisms of strong vigor. It means that organisms that are maybe resistant to diseases, to drought, to shortage of food, and all the like. Animals that are also very strong, that are sturdy, that do not just die easily. Okay? And cloning can also help in the production of organs as well, just like we did with stem cells. Stem cells, we can use them to genetically engineer new organs directly. Or we can use cloning to clone cells that are damaged. Just like we did, we produce nerve cells from stem cells. We can use existing nerve cells and we clone them and produce new nerve cells. That is called therapeutic cloning. But this kind of cloning that you see here, where an offspring is born, is called reproductive cloning. Other one would be therapeutic cloning. And this one here is reproductive cloning, where you have maybe like body the sheep. Explain why the nucleus of a skin cell was used in this process and not the nucleus of a gamete. The reason is because the nucleus of a skin cell, a somatic cell, is 2N. So it has the complete set of chromosomes. Meanwhile, the nucleus of a gamete, like an egg cell or sperm cell, is only N, it's haploid. It only has half the genetic material. So that is it. Okay, guys, we've come to the end of our long journey. I hope you enjoyed the lessons. All the best in your exams. Please make us proud. It was nice meeting you, even if I've not met some of you ever. But it was a pleasure. Bye-bye. Good night.
Bye bye.
If you have any challenges or anyone any challenges next year, you can contact me. I'll be available. So.